Hello, good morning, everyone. Today we will be doing lecture 2, which is macromolecules. And hope you have a chance to watch the recordings and download the study guide. We will be filling that in right now. Okay, so let's get started. First question, let's start with some basic chemistry. Okay, this lecture is kind of both biology and chemistry, kind of a little bit of biochemistry if we would. So we need to start with some basic chemistry knowledge before we can, do the biology stuff. So here, first question, list 5 major elements found in living things. So we are made up of many different types of elements, but there are five elements that are particularly high in percentage. Okay, so in no particular order, we have things like carbon. Okay, so the symbol for carbon is C. Okay. We have a lot of carbons. We also have a lot of oxygen. Symbol for oxygen is O. Hydrogen. Nitrogen. As well as phosphorus. Okay, so these are the 5 major elements that are common in all living things, okay, including humans. In addition to these major elements, we also have what we call trace elements. Okay, so trace elements means we don't have a lot of it. So these are only found in small quantity. Okay. Just give me a second. A student is trying to join the meeting. I'm just going to quickly send her the link. Give me 1 second. Okay, so these are only found in small quantity. In fact, if you have too much of the trace elements, it's usually going to result in some kind of disease condition. And in fact, if you don't have enough of these trace elements, you're also likely going to have some kind of abnormalities. Okay, so the key here is to have the right amount of these things, so that everything is about Goldilocks, right? About having the right composition. Someone asks, let me see, what is this question? What is the percent of phosphorus in our body? I don't have a number on top of my head. Let's see. According to Google, it is, let me see, phosphorus, we are talking about, sorry, a lot of things are blocking my screen. Phosphorus is about percent mass, we're talking about 1%, okay, 1% of your body mass. That's what Google says. Okay, so not a lot, certainly not as much as like carbon and oxygen and hydrogen, because phosphorus is primarily found in your DNA, right? Okay, so list 4 trace elements. Anybody want to give me some trace element, please? Yeah, Marlon said iodine. That is absolutely correct. Iodine. Okay, we need iodine for our thyroid gland. Okay, so the thyroid gland makes these hormones called T3 and T4. And they require iodine. Okay, these hormones regulate your metabolism. Zinc, Hashana says zinc, that's correct. Okay, helps with the defense. And then there is also magnesium, as well as someone said calcium. That's right. So the list goes on and on and on. Okay, but these are some trace elements that are found in living things. Moving on. Compare ionic and molecular compounds by completing the table below. So we can take the elements are everything on the periodic table. We can take these elements and we can combine them together. We can take a metal and a non-metal, and then they could combine together to form an ionic compound. You can take a non-metal and another non-metal and put them together, and you form a molecular compound. Okay, so the difference between ionic compound and the molecular compound, first of all, is the type of element that is being involved. So as I just told you, for ionic compound, it is going to be metal plus non-metal. Okay, usually it's one type of metal plus one type of non-metal. Okay, and then for molecular compound, it can be non-metals plus non-metal, and it doesn't have to be one type and one type. Okay, it doesn't have to be like, you know, NO2. Right? It could be something more complicated, like three things all together could form a molecular compound, like HPO4. Right, that's a molecular compound. The key thing is not so much the number of elements that are involved, but the type. Okay, so metal, non-metal gives you ionic. Now, in this type of a compound, the ionic compound, the bond that is being formed is what we call an ionic bond. So that's simple enough because it's basically the same name, right, as the compound itself. Okay, but what exactly is an ionic bond? Okay, an ionic bond involves the transfer of electrons from the metal to the non-metal. Okay, so you probably have learned this in chemistry class. Metals, they like to give off electron, right, or electrons to create a full valence shell, whereas non-metals, they tend to pick up electrons to create a full shell. So this is the perfect combination. When you have a metal and a non-metal together, the electron will go from the metal to the non-metal, and then they form an ionic bond. Okay. Whereas in the molecular compound, the bond is called covalent. Covalent bond. And this, rather than having a transfer of electron, it involves the sharing of electrons between the atoms. Okay. Any questions so far? To stay at room temperature. Okay, for ionic compound, because the ionic bond is actually quite strong. Okay, so at room temperature, these tend to be solid. Whereas for molecular compound, it could be solid, it could be gas, or it could be liquid. Okay, can anybody give me an example of a molecular compound that is liquid at room temperature? Anybody? Yes, H2O, that's correct. And water is liquid at room temperature. What about solid? A molecular compound that is solid? Okay, NaCl is actually ionic compound because Na is sodium, which is a metal, and chlorine is a non-metal. So that actually gives an ionic compound. For solid would be something like, for example, C6H12O6. Sorry, cannot really see that. We put it here, C6H12O6. That's glucose, right? And for gas at room temperature, we can have things like O2, okay? Or like CO2 even, CO2. So these are examples of molecular compounds that are solid, gas, or liquid at room temperature. Okay, so examples, I already gave you a bunch. But here, NaCl, right? You can table salt. And then another one is NaF. Anybody know where we find NaF? Probably used it this morning. I hope you did. No? This one is found in toothpaste. That's right, Tyler. Very good. And molecular compounds. It could be any of these things. I can give you a few more, like methane gas, right? That's molecular compound. Carbon monoxide, ammonia, all these things are molecular compound. Okay, if on the test I ask you, right, which of the following is a molecular compound, you can just pull up your periodic table and you can find whether these are metals or non-metals. If the whole thing is made up of non-metals, then you know it is going to be a molecular compound. All right, moving on. Indicate the polarity of a water molecule. Okay, so polarity refers to the overall overall distribution of electrons in the molecule. Okay, so sorry, many people are asking me for the link. You know what? They would just have to watch the recordings later. So the basic question you're trying to answer here about polarity is, are electrons being distributed equally, right, within the molecule? And if the answer is yes, that they are being distributed equally, okay, so if the answer to this question is yes, they are being distributed equally, then the molecule is going to be non-polar. And if the answer is no, the electrons are not being distributed equally, then the molecule is going to be polar. Okay, so we'll learn more about this in chemistry, but let's just look at water, okay, because that's the example we talked about in the recordings. So we can see water is H2O, 2 hydrogens, 1 oxygen, and the electrons are being shared in this covalent bond. Okay, but the sharing is not an equal sharing. Okay, even though they're sharing the electron, one of these atoms are actually kind of hogging the electrons more. It's like my kids. Okay, I buy them a new toy and tell them, you know, you guys got to share. I make sure you both have a chance to play with it. They are sharing, but then the older one always kind of hogs it. Right? I want to play with it more. Okay, so that's kind of like this. Okay, even though they're sharing the electron, the oxygen is actually going to be hogging the electron a little bit. So the electrons are going to hang around this atom more so than around the hydrogen, causing the oxygen to be slightly negative. Okay, and to write slightly negative, we use this special symbol. Okay, slightly negative. Okay, and the hydrogens are going to be slightly positive, like that. Okay, so how do you know which one is the one that's hogging the electron? Right, we look at something called electronegativity. Okay, and you will again learn more about it in chemistry. But basically, higher En, higher electronegativity, equals to stronger attraction for electron. Okay, so if you look up the electronegativity for oxygen, it's going to be higher than oxygen, so it's going to pull the electron towards itself. So that makes the water molecule polar. Okay, now it's important to note this. Even though the water molecule is polar, right, the overall molecule is still neutral. Okay, it's not like there is extra electron or missing electron. Okay, it's just that the electron is not being shared equally. Right? So you have one part that's slightly negative, one part that's slightly positive, but overall it is still neutral. Okay, so this point is important in case you bump into a question like that on the quiz. Okay, next question. Draw two water molecules and explain how hydrogen bonds are formed between them. Okay, so we have one water molecule here, and then I'm going to draw another water molecule right here. Okay, so oxygen slightly negative. Why is the color not changing? Oxygen slightly negative, hydrogen slightly positive. So just like magnets, when you put something that's slightly positive and something that's slightly negative, right, the opposite would attract. Okay, so this attraction that you have, that is the hydrogen bond. Okay, so I'm going to give you the definition of a hydrogen bond. Okay, and this is a simplified version. It's not entirely correct, but it will be sufficient for our purpose. Okay, so hydrogen bond occurs when the slightly negative part of one water molecule is attracted to the slightly positive part of, and I'm sorry, it's very crowded, of another water molecule. Okay. Sarah asks, how do you know it's positive or negative? Yeah, so like I told you that it is negative and this is positive, and you just kind of have to trust me, right? When you learn this properly in chemistry, your chemistry teacher, Michu, will tell you how to figure it out yourself. You look up the electronegativity basically, okay? But for our purpose, just trust me, okay? That oxygen is slightly negative and hydrogen is slightly positive. Okay. Any other question so far? Okay. Let's move on. Using a diagram to explain how ionic compounds such as NaCl are soluble in water. Okay, so NaCl, as I mentioned earlier, the Na is metal and the chlorine is non-metal, so it forms an ionic compound. And you should know, in general, ionic compounds can dissolve in water. Okay. How much you can dissolve varies, but generally you can dissolve some of it. Okay, so in NaCl, I told you the sodium, the metal, is going to have a positive charge because it gave away the electron, right? And chlorine is the one that's going to be accepting the electron, which would make it negative. Okay, so when you put it in water, you have the sodium here, that's going to be positive, and then you have the chlorine, which is negative. Okay, again, this is positive because it gave off an electron and the chlorine accepted the electron. So what is water going to do? Well, water, one part being positive, one part being negative, the negative part, which is the oxygen part, it's going to surround the sodium like that. Okay. Whereas for the chlorine, the hydrogen portion, the positive part, is going to be surrounding the chlorine. Okay. So as long as the water molecule can separate the molecule into like the positive and the negative ions, you will be able to dissolve them. Okay, so that's what it looks like. Any questions before I move on? So if this can happen, we call this molecule NaCl, we call it like hydrophilic, because it's water loving. Okay, something like a hydrocarbon, like a CH3, CH2, 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 CH3. Okay, something like this. This is called propane. Okay, sorry, this is called pentane rather. It cannot separate into positive and negative like sodium chloride. And as a result, it's not going to dissolve in water. Okay? It's going to be hydrophobic. Okay? Tyler asked, the NaCl are still chemically bonded together. Yeah, you can think of it like that. Okay, but as soon as you take away the water, they will go back together. Okay, just a little bit more chemistry. Compare acid and base by completing the following table. Okay, so we're just going to go with a very simple definition of acid. Acid is basically able to produce H plus when dissolved in water. Okay, so they can come in a little powder form. You add it into water, stir it up, it will produce these H plus. On the other hand, a base produce OH minus. Okay, we call this hydroxide, right? Hydroxide ion when dissolved in water. Okay. The pH scale is a scale that is used to measure the acidity or alkalinity of a solution. Okay, so if it's an acid, then the pH will be less than 7. Okay, so you have stomach acid, that's around a pH of 1.5. And then for base, the pH is going to be greater than 7. If it's exactly 7, then it's neutral. Pure water is neutral. Properties. Right? So acids, they are sour. Right? Think about like orange juice, vinegar, those are sour. They are corrosive. Okay. They can conduct electricity. Base, they are usually bitter. Okay. If you put a bar of soap in your mouth, taste it, it's bitter. Usually feels slippery and slimy. Also conducts electricity. Okay. Conducts. Example, I gave you some already. Vinegar, right? Orange juice. Any other example you want to give me? Anyone? Yep. Most of the soda, right? Pops, right? It's actually phosphoric acid in some of them, as well as carbonic acid. Yeah, coffee is coffee. It's a bit acidic after you leave it for a while on the counter. Base. What are some examples of base? Anybody? Yep. Bleach. Milk. Okay. Blood is actually a little bit basic. 7.35. All right. Moving on. Do we have any poll questions at the moment? Not yet. Okay, we're going to come back to it. Okay, list 4 types of macromolecules. So the ones that we've looked at in the lecture include carbohydrates. Right. The carbs, right? That's what we call them. We have proteins, lipids, and of course, nucleic acid. Okay. So the proteins, the carbohydrates, and the nucleic acids, these are polymers. Polymers of repeating units. Okay, not so much the lipids. Okay, the lipids are not not really polymers. They're not of repeating units. They are more like, components that are put together. So we'll talk more about that in a bit. So here is a diagram that shows you the relationship between the monomers. Okay, so monomers are individual building blocks. Okay, and they could be same, they could be different. For example, in proteins, you have 20 different types of monomers. For carbohydrates, it's more like 4 or 5. And when you link them together, they become polymers. Okay. I want to clarify, these are not the only polymers that exist in the world, right? Okay, these are just common polymers that are found in the body, right? There are many polymers that is not in living organisms and are useful in everyday life. Plastic is a polymer. You might be wearing a coat that says polyester, right? That is a polymer, right? If you cook with a non-stick pan, right? That coating that makes it non-stick, anybody know what that's called? Yeah, the Teflon, right? Teflon is a polymer. So many polymers that exist, but we are just talking about the ones that are found in the body. Okay. So to connect the monomers to form polymers, we are going to need to do a chemical process called dehydration. Dehydration synthesis. Okay. And other name for this is condensation. Condensation reaction. Okay, that is where you will remove a water molecule and connect the monomers together. Okay, and that's why there's a water molecule here. And when you want to break the polymers, after we consume the polymers for energy, you can break it down using the reverse process, which is hydrolysis. Okay? To break it with water. Hydro is water, lysis is to break. Okay. So let's start with carbohydrates. And again, if you have a question, please just interrupt me at any time. Okay, first chart here. The elements that are contained in carbohydrates. So we only have C, H, and O. Okay. Carbohydrate is carbon with water, basically. So the general formula is like CnH2ON. Right? So it means for every unit of carbon, you will have the same unit of H2O, hydrates in it. So example, if N is 6, then we have C6, H12O6. Okay, you put a 6 here, 6 times 2 gives you the 12, and then 6 times 1 gives you the 6, right? So that's basically your glucose, for example. Okay, so the building blocks, we have things like glucose. What's another monomer for carbohydrates? Anybody? Yep, fructose, that's correct. And I'm looking for one more. Lactose, not so much. Those are, people are saying lactose, sucrose, right? Those are actually no longer monomers. They are, when you put two monomers together, that's when you get those. We'll get to it in a second. Yeah, a couple of people say galactose, that's correct. Okay, so it's important to know which ones are the monomers, which ones are not. Okay, there's likely going to be a question like that on the quiz. Now these are isomers of each other. Okay, so same atoms, but arranged differently. Okay, it's like a small box of Lego. Okay, you can build a car, you can build a ship, you can build a plane. They look different, but they're made up of the same building blocks. Tyler asked, would monosaccharide be an acceptable answer? Yeah, so monosaccharide is the general term for these building blocks, right? But if I ask for like specific examples of monomers, then I would prefer you to include these ones. Okay. Biological functions. Why do we need to have what do we use carbohydrates for? Well, it is the main source of energy. Okay, you can burn it for energy. Okay, so the preferred source is glucose. Okay. We can also use it for storage. Okay, storage of energy. Okay. There are two types of carbohydrates that are useful for storage for energy. What am I referring to? And again, these are questions that you could expect, right, on a quiz. Which of the following is a storage form of energy? And then I can list, fructose, glucose, and then a bunch of other stuff. Very good. So starch for plants and for humans is glycogen. Okay. And one more biological function is structural support. Structural support, not so much for us, for humans. For animals, we don't really use, well, humans, okay, I should say. For humans, we don't use carbohydrates for structural support, but other organisms, they do, okay? For example, plants, they use cellulose, right? Cellulose, that's where the fibers are, what the fibers are made up of. And also for exoskeleton of insects, right? And crustacean like crabs and lobster and stuff like that, they have chitin, right? Okay, so these are the examples that are related to the biological function. Okay. I hope I'm not going too fast. Any questions thus far? I'm always concerned that you guys have questions and I don't give you an opportunity to ask. Okay, there is a question. Good. Is starch an example of a monomer? No. So starch is an example of a polymer. Okay. These are the only monomers that we learned in the lecture. Okay. So I'll get to the polymers part again later on, perhaps. Okay. In the following diagram shows how a bond is formed between two glucose molecules. There's a glucose molecule here. There's another glucose molecule here. You will not have to recognize these. I will not show you a bunch of these diagrams and ask you to choose which one is glucose. Okay. So you don't have to know how to draw them. You don't have to know, exactly what they look like. What you do have to know is that if I have two glucose molecules next to each other, what's going to happen is we are going to be taking an OH from one of the glucose, okay? And OH is called a hydroxyl group. Hydroxyl group. Okay. Now, just in case you're confused, earlier here, I said, what did I say earlier? Where is it? Earlier here, I said OH is hydroxide ion, and now I'm saying is hydroxyl group. Like, what am I talking about? How are they different? Well, this one is actually, sorry, this one is OH minus, right? Okay, that's what a base will produce when you put it in water. Okay, so with the minus, we call it like a hydroxide ion. And over here, if it's attached to part of a bigger molecule, we call it a hydroxyl group. Okay, so anything that has OH in it, right, we say it has a hydroxyl group. So there's a hydroxyl group, hydroxyl group, hydroxyl group, hydroxyl group. Okay, not terribly important, but just in case you wonder. So what's going to happen is you're going to take an OH from one molecule, you're going to take an H from another molecule, and then you are going to create a special bond that links them together. So like this. Okay, so you're going to create a bond that connects this C with this O, like this. They're going to connect together, okay? And you will form a covalent bond, a new bond. Okay, there's a name for this bond, but we're not going to care. So what is it going to look like afterwards? Well, I'm going to draw this hexagon looking thing again. Okay, there's oxygen here. Okay. There we go. Okay, so you can see this oxygen. This oxygen is this oxygen. Okay, and then this carbon is right here. That's the carbon. Okay, so this is the original glucose on the left, original glucose on the right. And after you remove a water molecule, you will be able to form a new bond. So this is a disaccharide. Okay, 2 sugar. 2 sugars. Okay. So don't get intimidated by these like huge structures. All I'm saying is to link them together, you would remove a water molecule. Right? And how do you do that? You will take an OH, a hydroxyl group from one, you take an H from another, and then you'll be able to form a new bond. So let's take a look at this question. What if I ask you this on the test? Quiz or whatever. Let's set up the poll. Okay, go ahead. Give it a try. Okay, I'm just gonna give it a couple more seconds. If you're not sure, you can choose E, so that way I know you are participating. Okay, very good. Okay, so that's the result, and it seems like most of you got it, right? So there is one molecule here with a hydroxyl group and then another hydroxyl group. And based on what I just told you in the previous question, you will take the OH away from this one. I'm going to take it away, and then you take an H away, and then you will form a bond like this. So you have the hexagon. The hexagon just means that there is like carbon on it, okay? Like, but minor details, not important. There's a hexagon, and then you're going to join the hexagon to the O, which would then connect to the pentagon. So that's the correct answer. Okay, plus the water, of course. So yeah. So there's a question actually, but I actually don't know which is different between B and C. Oh, so C here is 2 pentagons, right? Okay, so you want to connect like this guy, like number 1 to number 2, right? You see that? That's why it's hexagon to pentagon, not 2 pentagons together. That's the difference between B and C. Okay, and you have to have the oxygen, right? Okay, you have to have the oxygen. That's the key. That's why we don't choose A, right? And nobody chose A, which is amazing. Excellent. Are we good? Okay, so if you see this question, right, you know what to choose. Not necessarily B, okay, because it's randomized, the answers, and then you might get a slightly different diagram. But then the point is, you know how to find the answers, hopefully. Okay, What else is here? You know what? Let's do this one. Why not? I think we can handle this. Select all the false statements. Okay. So this one is a multi-select question. Go ahead. Okay, I'll give it another half a minute or so. Remember, these are potential quiz test questions, right? Okay, so you might want to take your phone and snap a picture of them every time they come up. You know, or watch the recordings again later. Okay, has been 2 minutes. I'm gonna end the poll and share the results. Okay, so all the false statement. Let's read all of them. First one: polysaccharides, which is the polymer of carbohydrates. Saccharides means sugar, right? Are important for both energy storage and provide structure to cells. We know that it's true, right? Energy is like glycogen and starch, right? We wrote down earlier, and structural support to cells is like the cellulose makes up the cell wall. So that is a true statement. We are looking for the false one. Glycogen is important for both energy storage and provide structure for cells. That is not true. Okay, glycogen is only for storage of energy, does not provide structural support. Cellulose is only found in animals. Not true. Okay, it's also it's actually found in plants. Okay, not animals. Okay, and it's not only in plants. Actually, you can find it in things like fungus and stuff like that too. Okay, but cellulose found in plants, not animals. That's the part that's wrong. The animal part. Glucose, fructose, and galactose are examples of monosaccharide. We list the monosaccharides earlier, right? Remember? So those are it. Finally, starch is the storage polysaccharide made by plant cells. That is correct. So the incorrect ones are B and C. Okay, B and C. Okay. All right, let's go back to the study guide. So in case I was not being clear here, I think Tyler should have mentioned that earlier. I should have wrote it down, but let's write down again. These building blocks, they are mono. Mono means single. Saccharide. Okay, single sugar. Single sugar. Okay, so remember I said like having a summary help sheet for the test, right? Maybe you need to write down what the monosaccharides are if you cannot remember them. Although I think you could manage. It's only 3. Right? Here we are going to complete the reaction. Okay? The maltose, the sucrose, and the lactose. These are all disaccharide. Disaccharides, double sugar, right? Yes, I will be posting this online afterwards. Okay, someone just asked. And where to find it, I'll show you afterwards. Complete the reaction. Okay, maltose is glucose plus glucose. Okay. What about sucrose? Anybody help me out here? Sucrose? Well, the good news is they all have one glucose, so that helps with the memory load. And for sucrose, yes, it is glucose plus fructose. Okay, and finally, lactose is glucose plus galactose. And this is probably quite easy, right? Because they both have the word lactose in it. Right? So again, these ones on the left, those are the monosaccharides, and then the ones on the right, they are the disaccharides. Okay. Moving on. Where can it be found? Okay, for these polysaccharides. Okay, so starch, we can find it in plants, right? Plants. Okay. So example, roots. Anytime we eat the roots of a plant, tons of starch. Think of potatoes, right? Think of like yam, right? Think of like carrots, tons of starch, right? They store it in the roots. And the main biological function of starch is to store, right? Store energy. Okay. So when it's the winter time, they cannot do photosynthesis as much. They can use the energy from the root. Okay. Store energy in plants. Glycogen. Okay, where do we find glycogen? We find it in animals. And such locations such as liver, muscle. Okay. Your liver is on the right side, just below your diaphragm. The diaphragm is right here, and then the liver is around this area. And what do we use for it? Store energy in animals. If you did not eat breakfast this morning, your liver will be releasing glycogen. Okay, and then you will chop it down into glucose so that you can have energy to go on, to carry on. Next one is cellulose. Cellulose is found in plants again. Okay, so example, cell wall. Right, for those of you who don't know what a cell wall is, this is a cell. Okay, cell for the plant is usually more rectangular. This is the nucleus, the inside of a cell. And outside of the cell, there is a little additional protective barrier for plants. That's the cell wall. And that's where all the fibers are. Okay. So what is it for? Structural support. Structural support. Okay. Maintain cell shape. Maintain cell wall, you can say. Last one is chitin. So we can say exoskeleton of insects, right? Or crustacean, right? Like lobsters, like seafood with a hard shell on the outside, right? And what do we use it for? Well, protection. What they use it for, rather, protection and support. You know, those supplements people take. You might know someone older who take these things, or you might take these things. The glucosamine, right? That's supposed to be good for your joints and stuff like that, right? They basically extract it from the shells of crustacean. Okay, it's based on chitin essentially. Okay, and supposed to be good for our joints. So there you have it. That is carbohydrates. Any question at all for carbohydrates before we continue on? 1 down, 3 to go. Okay, just a quick poll. I hope everybody get this one right. No pressure. But let's see. Okay, without looking, are you able to answer this question? Tyler asked for question number 10. Is water also a byproduct of each reaction? Yes, absolutely. I just didn't write it in the thing. But whenever you do dehydration synthesis, you are always going to get a water molecule. Okay, I'm going to end the poll. Majority of you got it, right? Glucose and fructose. Okay, for those of you who choose 2 glucose, glucose plus glucose is going to give you a maltose, and we are looking for sucrose. Okay, so that's not the answer. The correct answer is D. Okay, now that I have most of you here, let me just go to East Centennial and talk about a couple of things. Which is, hold on, right there. Okay, so when you go to the course homepage, eCentennial, again, right, like a lot of you email me on Monday asking me like, you know, where is the class, and you know, how do I get on the Zoom? We don't have classes on Monday, right? We don't have the synchronous class on Monday. Okay, you just go watch the recordings and stuff like that. So every at the beginning of every week, usually I post it on Sunday, right? On Monday, there is going to be a to-do list. Okay, so you just log on to eCentennial, and then I put all the links here for you to click on. You don't even have to find the stuff. Okay, you just click on it, and then it will take you to the lecture 2 recordings folder, and you will find the PowerPoints, and you just watch these things. Okay, so you don't have to email me for the links. I will post it as a news item at the beginning of every week. Okay, so this week obviously we're doing lecture 2, and then you also have to study for quiz 1. Okay, and here are the self quiz. If you haven't already had a chance to try them, please do, because it will also help you familiarize yourself with how the quiz is going to be like on the actual test, right? Like in terms of like the technology piece, right? How the interface is going to be. So please, please do that. And there is a question. And where to find the link? What link? The link to what? I think you have cap lock on as well. The links to what information are you looking for, Niam? Okay, I'll wait. To the course. Oh, like how to get the homepage here? Oh, okay. There. Okay. There it is. But oops, sorry. I send it to here. That's the link, okay? So it's where you find all your other courses, right? Basically, if you go to eCentennial. But in case you didn't know, that's what it is, and you can log on with your credentials. Anyways, the quiz next week, guys. The quiz next week is going to be available on Monday, right? Monday from 6 a.m. to 11:59 p.m. Please do mark it down on your calendar. It's going to be 45 minutes, and it's on lecture 1 and 2. And you can click on the link here. It will take you to the quiz. If you click on it now, you won't have access to it. Okay, it will only open between those times. It's the first quiz, so I try to keep it light. 20 regular multiple choice questions, 5 multi-select, no application yet. Okay, we'll have the application questions later on, and this is going to be 10% of your final grades. Again, if you register with CALS and you need accommodations, please email me the documentations no later than Friday, so I can set you up properly. Okay. Tina asked, will there also be a lecture recording for Monday or just the quiz? Yes, there will be lecture recordings. You will have to watch lecture 3 recordings. But like I said, the recordings, all the recordings for the entire course is up already. So if you know you're going to be busy on Monday doing the quiz, you can watch lecture 3 recordings like today or tomorrow or Friday. Okay, you know, make your own schedule work. Finally, okay, as you might or might not know, at the end of each class, I will post a debrief, okay? And it will say you can find the study guide answer, right? Which is the stuff that I'm writing as we do it, right? You can always find it here. You just click on this link, study guide answer, right? And you can see the lecture 1 there. So today after class, you will see the lecture 2, okay? And then so on and so forth. It's the same link every time, right? One of you were asking me where to find it, right? And then there's also the tutorial recordings, right? Like I said, you click on it and you just bookmark it. Then you can watch them if you missed anything. If you come in late or if you have to leave early, or if you miss it entirely, that's where you can find it. Okay. And the Zoom link is the same every time. Okay. I got a few emails this morning asking me where to find the Zoom link. Here it is. Bookmark it, put it in a safe place. Okay, let's go back to our stuff. How do I do that? Here, I think. Yep. All right. Next, we will have lipids. Okay, lipids. So one key thing to kind of note for lipid is, you know, unlike the other 3 macromolecules. Okay, so unlike carbohydrates, proteins, and nucleic acids, lipids are not all made up of repeating monomers. Right? Instead, they consist of various, we'll call them components, okay? Components. Okay, so first here, we have different types of lipids. And the first type of lipids that is shown here is what we call a triglyceride. Triglyceride. Which is just a fancy word for fat. Okay. So triglyceride or fat has 2 major components. Okay. We have this molecule right here. Okay. That is called a glycerol. Glycerol. And then we have these stuff right here. Okay, these are the fatty acids. So 3 fatty acids. Again, you don't have to know what they look like chemically. You just have to memorize them. Okay, so we'll select all the components of a triglyceride. Okay, so you would choose the glycerol and the fatty acids. Okay. So what is the main function? Okay? Or what are some of the main functions of fat molecule? They can store energy. Right? So we store the excess glucose as glycogen in the liver and in the muscle. But if you consume way more than you need, some of it will be converted into fat. Yeah? So we store them as energy, right? Long-term energy. So as I said, if you didn't eat breakfast this morning, you'll be burning your glycogen reserve. Okay? But if you continue to not eat for the rest of the day, and then you don't eat for the next day and then the day after, then you will start to tap into your fat reserve. Okay, in fact, that process might start, after 16 hours or so. You start burning your fat, right? Which any of you have heard of that diet thing? This is like a new trend. My parents are doing it. Some of my friends are doing it. They're like, you got to starve yourself for 16 hours, 16-hour fasting, right, before you eat, right? Because like 16 hours is supposed to be the golden time where if you don't eat, your body is going to start to burn your fat reserve, right? Okay, so those are long-term energy storage. Okay, we also need a little bit of fat to be healthy, because they do insulation. Okay, insulate and protect organs. Okay, you might have a little bit of fat around your heart, around your kidneys, your major organs. So when it's like minus 33 degrees outside, your organs are going to be, nice and warm because of the fat insulation. Okay, so there you have it, triglyceride, fat. Next one. Anybody know what this one is? Okay. We also see the glycerol again. Okay, we have two fatty acids. And we also have a phosphate head. So that makes it a phospholipid. Phospholipid. Okay. So number one, we have the phosphate group. Phosphate group. And this is the part that makes it polar. Okay. And then the tail, the fatty acid tail, that makes it non-polar. Okay, so this is a special molecule. It has a polar part as well as a non-polar part. Okay, so number two here, we have the glycerol. And then number three, we have two fatty acids. Phospholipids, they are the major component of cell membrane. Again, here's your cell with a nucleus. The membrane is what surrounds the cell. It separates the living stuff on the inside and the non-living environment on the outside. So this cell membrane is primarily made up of phospholipids. Okay. And again, this molecule is special, and usually we draw like this. Okay, we have the head, and then we have the tail. Okay, the head is polar, so it is going to be hydrophilic. And then the tails, they are hydrophobic. Okay, so the head will interact well with water, but the hydrophobic part will not. Okay, the hydrophobic part will stay away from water, and that property is going to be important when we discuss the cell membrane in more detail later on. Any question about this molecule and its relationship with hydrophilic, hydrophobic, and stuff like that? No question. Okay, let's finish the next one, and then we will take a quick break. Okay, I know you guys must be tired from hearing me talk for an hour. What is this one? This one has a bunch of rings in it. Okay, 4 rings. Anytime you see 4 rings fused together like this, we know it is going to be a steroid. Okay, a steroid. Okay. So a steroid molecule has these rings. Okay. We call them carbon rings. So 4 fused carbon rings. And so if I show you a picture of this, you see 4 fused carbon rings, you know we're talking about steroids. Okay. And number two, we're gonna have all these like almost like accessory that we stick onto it. Okay, so these things that we attach onto the rings, they are called functional group. Functional group. Okay, and these functional groups, think of them as like add-ons, okay, or accessory to your phone or whatever. Okay, they give it unique properties. Okay, so here again we see the OH, right? Remember OH, the hydroxyl group, and you can stick other stuff onto it as well. So an example of a function for steroid is hormones. Okay, not all our hormones are steroids. Right? We have two classes of hormones. We have the protein hormones, such as like insulin, for example, and then we have the steroid hormones. Okay, one is protein-based, one is lipid-based, and they function differently. So examples of steroid hormones would be the sex hormones. Anybody know the sex hormones that we have in the body? The main male sex hormone and the female sex hormones, what are they? Yep, the testosterone for male, and for women, it is estrogen. And the other one, what's the other big one for female? Anybody know? Yes, progesterone is correct. Okay. Men also have these, right? Just at lower levels, vice versa. Okay. These are the dominant one for male, dominant one for females. Steroid, it might be surprising. It also forms the water repelling surface. Repelling surfaces. Like the waxy coating, right? It's steroid as well. Okay? And a lot of people think steroids, they think of like performance enhancing drugs, which is correct. That is a type of steroid, but that's not the only kind of steroid, right? Okay? Cholesterol is a type of steroid. Okay? And we need cholesterol because cholesterol is also part of your cell membrane, and the cholesterol also is the starting material for testosterone and then estrogen and progesterone. Okay, so don't think cholesterol is like bad, right? Too much of a good thing is a bad thing, but not necessarily all bad. Are we okay together? Anything before we take a quick break? Okay, let's take 5 minutes break. Okay, so now on my clock is 9:37. We'll come back at like 9:42. Okay, 5 minutes. Okay, I'll see you in a bit. Hello again, and we will continue. Next question, number 13. Compare saturated and unsaturated fat by completing the following table. So saturated fat, in case you're confused, we're talking about the different types of lipids. So there's fat, there's phospholipid, there's steroid. And then for fat, there are two types of fat, the saturated fat and the unsaturated fat. Okay, so what's the difference? Well, for saturated fat, it is going to be 1 glycerol plus 3 fatty acids. Okay, and the type of fatty acids that we will find in the saturated fats, well, as the name implies, they are going to be saturated. Saturated fatty acids. Okay, so what does that even mean? Well, saturated fatty acid means that the carbons in the fatty acids are bound to max number of hydrogens. Okay, you cannot fit any more hydrogens onto it. Okay, it's maxed out with the number of hydrogens. Okay, and as a result, they are usually straight in structure, no kinks. Okay, no double bond. Sorry, I always have trouble allocating the proper space on the worksheets for students. So do apologize. Okay. So if I have to draw the fatty acids in a picture, they would be there. There is your glycerol, maybe. Okay, glycerol. And then you will have the 3 fatty acids. They are straight. There are no kinks in it. And for that reason, they can pack really, really close to each other. Okay? They can pack really close to each other like that. Okay. And for that reason, because they're closely packed together at room temperature, they will be solid. Okay? Solid at room temperature. Can I have some examples of saturated fats that you will find at home, maybe? Yep, butter. Okay, so example, butter. Lard. Okay. Yeah, coconut oil. Coconut oil, so good, but seems so fat. Okay, so these are generally derived from animals. Not always. Okay, like coconut oil is obviously not derived from animals, so it's an exception. But generally, they are derived from animals. Okay. Unsaturated fat, on the other hand, let's choose an exciting color. Unsaturated fat, we have 1 glycerol plus 3 fatty acids. So it's the same thing. It's the same thing. However, the type of fatty acids here we have would be at least 1 unsaturated fatty acid. Okay, you can have more than 1. You can have all 3 of them being unsaturated. Okay, so what that means is, well, it's the opposite of this, right? So for this, the carbons are bound to maximum number of hydrogens. So in this case, the carbon, some of the carbons are not bound to max number of hydrogen. Okay, so there are rooms for more. Have you heard of this term before? Hydrogenated fats. Hydrogenated fats. Anybody heard of that before? Right. Things like shortening, right? That's an example of hydrogenated fat. That is when they take an unsaturated fat and they chemically add more hydrogen to it, and then it causes it to become like a more solid. Okay, like margarine, right? You know that stuff. What's the slogan? Can't believe it's not butter. That's hydrogenated fat. Right? Very unhealthy in a sense, right? Because you chemically convert a liquid type of fat into a solid form, so you can spread it on your toast, right? And that is hydrogenation. Okay. Yes, can't believe it's not butter, because it's not. So this is rather than straight, it's going to contains kinks, maybe more than 1 kinks. Okay, some double bond. If you're not sure what a double bond is, I'm not sure if you learned that in chemistry yet. But usually, you know, in water, right? In water, there's 1 line that connects the O and the hydrogen. That's a single bond. There's like sharing of 2 electrons. sometimes you can have like a double bond like that. So that means instead of sharing 2 electrons, they're sharing like 4 electrons. Okay, that's all it means, to keep it in the simplest form. Okay, so next, because it has kinks in it, let me try to draw that out for you. So you would have your fats, and then you might have like 2 saturated and then 1 unsaturated, right? It's kind of like sticking out your legs while you're trying to line up, you know, in a place. With this sticking out of the tail makes it very hard for the molecules to pack together. Okay, you may even have 2 unsaturated fat. Okay, so because they're not compact together like the saturated fat, they're kind of spaced out, right? These tend to be liquid at room temperature. Okay, so example would be like your cooking oil, right? So like I don't know, like olive oil, right? Canola oil, right? Yeah, vegetable oil, right? Avocado oil, whatever you use. What does it mean to have a kink? Yeah, so this is the kink, right? A kink means like a bend, right? There, a kink, K-I-N-K. All right. So basically, again, for those of you who want to know a little bit more, if you have just single bonds, the fatty acid looks straight. If you have a double bond, okay, it will cause it to bend. Okay, so that double bond right here, right, that creates the sorry, it should look like this. There's the double bond. Right? So that creates the kink, right? And it's because of the kink that they're not able to pack together. Okay, again, think about like lining up single file instead of like, you know, having your hands straight down. You know, stick your hands out, you spread your feet out, and you try to, you know, have your social distancing, right? Keep people away from you. That's basically what the unsaturated fat is like. Okay, they don't pack together as closely as saturated fat does. That's why they're liquid at room temperature. Okay. So which one do you want to consume? Which one is healthier? Unsaturated fat tends to be healthier than saturated fat. Okay, but again, too much fat is obviously not good either. That is it for lipids. Any question about lipids or like, you know, carbohydrates before we move on to proteins and nucleic acid? Proteins is the one that is intense. A lot of stuff going on with the proteins. We all good? All right. Proteins. Label the different parts of amino acid below. Okay, so in case you forgot, amino acids, they are the building blocks. The building blocks of proteins. Okay. So that's what a typical amino acid looks like. Okay. There is the central carbon. Okay. There is the amino group right here. Okay. This NH2 part, that is the amino group. And you do have to know the name, okay? Amino group, the NH2. And on the other side, we have this thing, the COOH, and that is the carboxyl group, which is also known as the acid part. Okay, and we have the R right here. The R is going to be different for each of the amino acid. We call that the side chain. Okay. So side chain is unique for each amino acid. Okay. Everything else is the same for the amino acid. They all have a central carbon. They all have an amino group. They all have a carboxyl group. And then they all have a hydrogen here. Okay. So just like everything else we talked about so far, we can take the monomers and we link them together to form polymers. Okay? And we link them together by removing a water molecule. Okay? So what are we going to do here? We are going to, pay attention, we're going to take away the OH from the carboxyl group, and we're going to take away an H from the amino group. And they're going to come out, and we will have H2O. Okay. So if you add these two things together, you can remove a water molecule to connect them together. So what will it look like afterwards? Okay, this part is going to look confusing. You don't have to draw it. You can just watch me, and then I will upload this afterwards, right? So we will have the CH NHH, or I'll call it R1, right? So we know we're talking about the first one here. And then we will have C double bond O. And then the new bond, the new chemical bond, is going to connect this carbon with this N. Right? That's going to have a new bond right there. I'm going to draw the new bond in pink. Okay. And then we will have the N. We will have the N and the H. R2. Okay, so this new bond is called a peptide bond. Peptide bond. Okay, and you form the peptide bond by connecting the carboxyl group with the amino group together. And that's how you get two amino acids to join together. Okay, and this could keep on going and going. Okay, I'm just going to show you a few more because I have a point to make. Okay, so this one, we're going to remove the OH and we're going to connect it. We're going to have another peptide bond. I'm just going to draw in black this time. And then we'll connect to the amino group of the third amino acid. And then it will connect to the amino group of the fourth amino acid, and so on and so forth. And you would have hundreds and hundreds of these things connected together, right? And then so on and so forth. Okay, so what you can see here is you will end up having a backbone that is essentially the same thing. So look, right? NHCH, C double bond O. NHCH, C double bond O. And then again, NHCH, C double bond O. And then it's just going to be repeating. And then it's going to be keep on going and going and going. And it's going to form your polypeptide. Polypeptide. So polypeptide is the equivalent of polysaccharide, right? It's like the protein version of polysaccharide. Many, many amino acids linked together. Okay. Excuse me. And the backbone, the peptide backbone, I'll label it for you. Right? The peptide backbone is the same. It just keeps on repeating itself as I've shown you. Right? What is different would be the side group, right? The side chain. Depending what side chains you have, it's going to give your proteins different properties. Okay. So that is that. All right, let's talk about the elements. What do we have in proteins? We have the carbon, the hydrogen, nitrogen, oxygen, and actually plus traces of sulfur. Okay, so sulfur is found in one of the amino acid. Okay. So how many amino acids do we have in our body that makes up the protein? How many building blocks do we have? Anybody remember that? Yes, we do have 20 amino acids. 20 amino acids, right? 12 of them are non-essential, and 8 of them are essential. The essential ones you have to consume, you have to eat it from your diet. The rest you can just make yourself. Biological functions. They could be enzymes. Okay? And enzymes, they are important for speeding up chemical reactions. We need proteins for structure. Your fingernails, your muscle, your hair, they are made up of proteins. We need proteins for hormones, as we mentioned earlier. Some hormones, lipids, others are proteins, so such as insulin. Okay, you might have heard of insulin. You might have heard of oxytocin. That's the one that helps in labor, right? Push the baby out. And in very, very extreme cases, if you're starving, fasting for a very long time, you will burn your fat reserve, and then you will also start to burn your protein reserve. Okay, so these are the main biological functions. All right. Next, complete the table about the structural levels of proteins. Okay, the first one is already done for you. Our primary structure refers to the sequence of amino acid that we link together. So which of the 20 amino acids do we put together? Well, this is determined by the DNA code. Okay. And it's basically like, you know, the first amino acid could be like alanine, and then you connect it to another one like a glycine, aspartic acid, you know, whatever the case. Okay, so the order of amino acid, right? And you can repeat them, right? It's okay to repeat them sometimes. Okay, so this order of amino acid, this chain, if you read the identity of each of the amino acid from one end to the other, that's the primary sequence. Okay? And what holds these amino acids together? The peptide bond that we talk about, right? The C double bond O and the NH, like that. Okay, so that is the peptide bond. Secondary structure. Okay, so remember I drew the peptide bond earlier? The backbone, rather, I'm sorry, the backbone, right? This is the backbone. So what happens is the backbone sometimes will curl up into these spiral, right? We call those alpha helices. And then some parts will form these zigzag patterns. Okay, so whether they form the spiral, the helix, or the zigzag, that is what we call the secondary structure. Okay, so we have alpha helix or beta sheets. Okay. Beta plata sheets, which is called beta sheets. So here are the beta sheets. Here are is the alpha helix. Okay. So do they form the helix or do they form the sheets? That's what secondary structure means. Okay. And how do we hold them together in the secondary structure? Okay. The helixes and the sheets, they are held together by hydrogen bonds. Hydrogen bonds from the backbone. Okay, hydrogen bonds from the backbone. So let me just quickly go back here and explain it briefly. Okay, so just like water, right? The oxygen is slightly negative and the hydrogen is slightly positive. So if I continue this backbone here, it's just going to be the same thing over and over and over again, right? Okay, so I might have an other CH here, and then that will connect to like a C double bond O, and then an N H, and so on and so forth, right? Keeps on repeating. So over here, that's slightly negative, the oxygen, and the hydrogen is slightly positive. So there will be some attraction here. There will be some attraction like here. Right? And overall, these hydrogen bonds, it's what's going to stabilize the helix and the plata sheets. Okay. Again, don't worry too much about how to draw them. This is just for people who are kind of interested. But yeah, so you will have the hydrogen bond like here, stabilizing the structures. Think of them as like little toothpicks that, support the structures. And then afterwards, the protein will fold up together to form a three-dimensional shape. That would be the tertiary structure. Okay, so you would have the protein folding up like this. Oops, I just erased it. Okay, they might fold up on itself and form like a three-dimensional shape. Okay, so the overall 3D shape, overall 3D conformation, that means shape, that's what we call the tertiary structure. Okay, and what holds it together is the interaction between the side chains. Okay. And then some proteins will have quaternary structures. Okay, quaternary structures are when more than one, okay, two or more polypeptide come together to form a complex. Okay, so an example of that is hemoglobin, which is the protein that transports oxygen in our body. So they would have two black subunits and two red subunits, for example, and it will come together to form a complex. Okay, and what holds them together is the interactions. Interaction, more than one type interactions between the polypeptide. Okay. Any question? All right. Let's do a quick poll. Okay, I think we're just gonna skip this one. We don't have a lot of time left. What is a chain of 25 amino acid called? The answer is polypeptide, right? Okay, we talked about that already. Polypeptide. How about this one? Okay, let's do this one. Go ahead. Okay, looks like most of you got it. The answer is carboxyl group and amino group, right? Okay, and to find the answer, you just have to look at the notes, right? Carboxyl group and amino group. Very good. Let's try one more. This one, maybe we'll skip. Hemoglobin is an important protein, which is what we just talked about, right? So it has 4 peptide chains together, and the level is the quaternary structure. Okay? We just talked about it. Okay, so if you want to revisit this question, just watch the recordings after. We will do this one. Go ahead. Okay, a couple more seconds. Okay, looks like the popular choices are B and E. Okay, the key words here is protein. Okay, so pancreatic cells make insulin, which is a type of protein. That's the key word here. Okay, so these cells uses something in order to synthesize insulin by the process of something. So if we are talking about proteins, how do we make proteins? We make proteins by connecting amino acids together through dehydration synthesis, right? Okay. Fatty acids and glycerol, that's lipids. That has nothing to do with the proteins. So the correct answer is E. I hope that makes sense. All right, let's move on. Okay, so in the last couple minutes here, we will finish up with the nucleic acid. Okay, the nucleic acid. So first thing is the chart again. Elements contained in nucleic acid. So we have everything: carbon, hydrogen, oxygen, nitrogen, as well as phosphorus. Okay. The building blocks for nucleic acid is something called nucleotide. Nucleotides. Biological function. Well, it contains genetic information. And we use that genetic information to make proteins. Okay. Instructions to make proteins. Okay. How do we know what amino acids to connect to what amino acids? We need the genetic information to tell us that. Okay. So we'll learn about this in lecture 7, I believe. So here is a nucleotide, the building block for nucleic acid. Okay. And we have 3 groups here. We have over here a phosphate group. Phosphate group. Okay, it has a negative charge, so it's going to make the DNA negatively charged, the backbone anyways. We have a sugar here, and this is a sugar. And it might be missing an oxygen, it might not be. If it's missing an oxygen, we call it a deoxyribose. And if it's not missing an oxygen, it's a ribose. Okay, so deoxyribose in this case, okay, or ribose, sugar. And this case, because it's missing an oxygen here, it's deoxy. So this is in DNA. This is in RNA. Next, we have this little thing here on the side. That is a nitrogen base. Nitrogen base. Okay, could be A, C, G, or T in DNA, or A, C, G, and U in RNA. And more on that later. Identify each base as purine or pyrimidine. Okay, so these 3 things, these 3 here, these are the pyrimidines. Pyrimidines, they have 1 ring in their structure. 1 ring. Whereas purines are these ones. They have 2 rings. Okay. And in order to remember it, you can use this phrase, pure as gold. Okay, so purines are A and G, adenine and guanine, pure as gold. Okay, so instead, in terms of the pairing rule, right, pyrimidine always pair with purine. Okay, so A will pair with T and C will pair with G. Okay. Or U, I guess we can say. Okay. Let's compare the DNA and the RNA. The nucleotides involved would be phosphate, plus deoxyribose is just a sugar that's missing an oxygen, okay, a 5-carbon sugar, plus nitrogen base. And the base could be A, C, G, or T. And it's going to be a double helix. Okay, so it's going to look like this: 1 strand, 2 strands, and then you will have the base pair in between. It's like a twisted ladder, as they say. For RNA, we are going to have the same thing: phosphate plus a ribose sugar and also a nitrogen base. However, instead of the same basis as DNA, we have A, C, G, and uracil. Okay, so uracil replaces thymine. No thymine on RNA, only uracil. And it's usually as well, it is single strand. Yeah, so like that. It's possible for the single strand to fold up on itself to form various structures. Okay, so this is something called tRNA. Right? The RNA would fold up on itself and it's able to form bonds with itself. So we'll talk more about these things later on. Any question? Okay, let's do one more poll before we go. Let's do this one. Yeah, this one looks complicated. Multi-select. Okay, maybe half more minutes. Abdul asks, are we going to take up the long answer questions? No, we don't have time for that, but I'll post the answers. Okay. All right, let's see. We are looking for something that's not a feature. All right. So first one, nucleotides with a single strand are connected by a covalent bond. So the nucleotides are like the, you know, the phosphate sugar and the nitrogenous base, right? They are connected through covalent bonds. That is a correct one. Okay, I see that a lot of people chose this one because you see the word single strand, right? So what this is saying is within one strand, okay, this is the other strand, right? Within this strand, it's made up of nucleotide, right? So nucleotide, 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 nucleotide. What's connecting the nucleotide together? It is the covalent bond. Okay. In fact, everything we talk about today, you know, the sugar and the sugar, that is a covalent bond. The peptide bond is a covalent bond, right? What connects the glycerol and the fatty acid? That's a covalent bond. Every bond that connects the monomers together is a covalent bond. So maybe you should write that down. Okay. All bonds connecting monomers together are covalent. Okay, write it down, please. Next one, adenine forms a base pair with uracil. There is no uracil in DNA, right? So that is not correct. Okay, no uracil. This should be thymine. Complementary strands are held together by hydrogen bonding between the phosphate group. Okay, phosphate group, the backbone. Again, it feels like I didn't talk about this in what we just did, but these are covered in the lecture notes, okay? So backbone is phosphate plus sugar, okay? And if you think about, again, the two strands and then how the two strands are held together, and these are hydrogen bonds between the bases, okay? So this is the nitrogen base. Okay, so that would also be incorrect. And that's what we're looking for. We're looking for things that are not a feature. Lastly, complementary strands are anti-parallel running in opposite direction. I believe there's a picture in the PowerPoint that shows this, right? One goes this way, the other goes the other way, opposite, even though they're parallel. So we call them anti-parallel. Okay, so check the notes. And you will see what I mean by that. So the correct answer for this question is B and C. So unfortunately, we don't have time to go through these things. I will post the answer key for the long answer question as well as for the fill in the blanks. And you know, if you're not sure about the long answer questions, even after you look at the answer key, feel free to email me for clarification. So thanks again for coming. Please remember there's a quiz on Monday, and I will see you next Wednesday at the same time. Okay, have a good week, guys. Bye.